Thank you very much, Lumen, and thank you all for coming on a Tuesday. I hope you are not so sleepy because we are going to talk about more cool ferns. So we are going to talk about Parablechnum, but before I start, I would like to ask a question that is, doing taxonomy in the 21st century, why? Well, as you all know, the taxonomy is the science of classifying living organisms based on their characteristics and the evolutionary relationship between different species. And it's a really cool science and a really innovative science that involves a lot of different disciplines like morphology, anatomy, chemistry, molecular biology. However, when people think about taxonomy, sometimes they think about black and white pictures of old men with long beards, and they don't look really happy, to be honest. There is this wrong perception as taxonomy as an old-fashioned science. And this is concerning because taxonomists get fewer citations in their works. We usually publish in lower impact factor in this really unfair system of publishing. We get less funding, so we are in danger of extinction. Additionally, this is really concerning because there is a really important job that taxonomy do that is becoming less common, that it's monographs. This is a definition of a monograph that I like by my friend and colleague Pablo Muñoz. A monograph is a complete systematic work at a global level of a taxon carried out through the study of the organisms in its natural habitat, if possible, herbaria material and lab data. As I said, this is a really important job, but it's also a really difficult job to do. And I wrote you an example about a scientist that made a monograph. You may know him. His name is Charles Darwin. He made a monograph of barnacles. And at the beginning of his work, we could read in his letters things like, how I shall enjoy getting back to town with renovated halls if such is to be my good fortune and resuming in the beloved barnacles. But when we move forward a little bit on his work, what we see in his letters is the classification of barnacles is in perfect chaos. Literally, no one species is properly defined. The subject is heartbreaking. And if we go when he was doing the second volume, we could read, I am at work on the second volume of Theoripeda, of which creatures I am wonderful tired. I hate a barnacle as no man ever did before, not even a sailor in a slow sailing ship. So I think that all the people who worked on monograph can relate to this, but I insist, monographs are important, and actually Darwin finished the monograph of barnacles, and it has more than 1,500 citations since its publication. So monographs are important for organizing the existing information, re-identifying and possibly extending the specimens, describing new species, making new discoveries, and of course, more publications. And this is even more important in the time we are living in, because we are facing a biodiversity crisis. As you all know, the biodiversity crisis is an accelerated loss of genetic variety, species, and ecosystems. Indeed, 750 animals and 120 plant species become extinct since the 17th century. And also, right now, there are more than 18,000 plants and animals in danger of extinction. Plus, it is estimated that 86% of the species are still undiscovered. So what we get is that we are losing species before we even discover them. For these reasons, I believe that the taxonomic work is so important. But we have another problem. I'm depressing you guys. I promise this is getting better later. The other problem that we have is that there are bias on the biodiversity and the study of the biodiversity. This is a study that I like from Tourette and collaborators in 2017. And what they did was to look at the records on the GBIF platform, and they compare the total of records, the percentage of records in that platform of different group of organisms, and they compare that with the total percentage of the global biodiversity that their organisms were. So, for example, well, they set two parts. The green part is occurring success, and the red part is occurring so far. And for example, the group of organisms that is leading the green part, which are birds, are the 55% of the records in GBIF, even though they are only 1% of the global biodiversity. And this is really convenient because today we are going to talk about the organisms that lead the red part, Polypodiopsida, or ferns. I don't have to convince you guys why ferns are so cool. Sarah did it already so nicely. As he said, we have about 12,000 of species all over the world. And if you see some numbers of the number of species of several countries, you can see that most of the diversity is on the tropics, but they can live mostly anywhere. They are present in all of the continents except the Antarctica, and in very different environments, from deserts to rainforest. And how did they get this distribution? Well, we have a scenario really simplified of spores versus tectonics. On the one hand, they can reach really long distance, dispersing the spores by the wind. So some species or some clades got their current distribution by events of long distance dispersal, and some others by vicariance. They were in a place which became split in two places for events like the tectonics. So what is thought within ferns, again simplifying it a lot, is that first lineages got their current distribution by vicariance and younger lineages by long distance dispersal. So whenever we are studying a group of ferns, we should consider both scenarios. And this is a work that I like by Weston Testo and Michael Sundu, in which they did the different fern families and they calculated their speciation rate. And what they saw is that, as you can see here, the Blechnaceae and the Onocleaceae would be the most recent fern families. And also, the Blechnaceae is one of the families with the highest speciation rate. And you are so lucky because we are going to talk about Blechnaceae today. So where are we with the Blechnaceae? Well, this is the global phylogeny of ferns. We are here in the group of Eupolyploids 2. The family Blechnaceae has about 265 species. And they look like this. As you can see, they are beautiful plants. They have a great variety of habits. They can be terrestrial, epiphytic, saxiculous, even subarborescent, as Lomaria scomburkii. And you can see them easily in the field because they are reddish when they are growing. It's not unique to the family, but it's really distinctive. They have continuous sori on both sides of the costa. They are lineal. They are not always continuous. In some genera, as Woodbardia, they are discrete. And one fun thing is that within this group of Eupolyploids 2, one common feature is that they have two vascular bundles in the petioles. But Blechnaceae is the only family in this group that has more than two. This is the distribution of Blechnaceae among the world. As you can see, they are almost everywhere. The highest diversity is on the tropics, but there are some genera that appear in temperate zones. Here in the States, for example, you have Struthiopteris, Ankistea, it used to be Woodbardia, etc. So, the taxonomy history of the Blechnaceae is that we used to recognize 9 to 11 genera, of which Parablechnum comprised the 80% of the species of the whole family. But several studies suggested that this genus was indeed polyphyletic. So in 2016, Gasper and collaborators made a global phylogeny of the family and split it in 24 genera, with the addition of Spicanthopsis in 2019 by us. I am sorry about that. So this is a summary of the global phylogeny of the family. As you can see, these 25 genera are in 3 different subfamilies. And if you look at the number in brackets, it's the approximate number of species. So right now, the most diverse genus could be Parablechnum, which is the genus that I study. This is Parablechnum audiens Parablechnum. They are usually big terrestrial plants, with some exception. They can be also small, as this Parablechnum procerum or Saxiculus, and they have this cool feature of being red when they are growing. They are usually dimorphic plants, as you can see here in the Parablechnum sideanum. That means that the fronds that bear the spores are different from the ones that doesn't. They are one pinnate with a stalked pinnate, and they usually have this the structure that is pointed with an arrow that are called aerophores. And they are usually really scaly plants, not always. This is the distribution of the genus. As you can see, it has 2 hotspots of biodiversity, one in Central America and South America, and the other in the Austro-Pacific, with some species also in South Africa, Madagascar, and the Mascarenes. And it's a really difficult genome taxonomically. Why? First of all, because there are some species with a restricted distribution and or low number of collections. For example, Parablechnum howeanum is endemic to the island of Lord Howe near Australia. Parablechnum punicium has only been collected twice in New Guinea, or Parablechnum gemascens, which is only known by the type in Peru. Also, as in so many other groups, there is controversy about the number of species. For example, this is what we call the Parablechnum cordatum. Some species belong in the Parablechnum cordatum complex. There are some authors that consider that we have in Mesoamerica Parablechnum sideanum and Parablechnum falciforme, which is not there. Parablechnum cordatum with a wide distribution in South America, and Parablechnum chilense, endemic to Chile and Argentina, and some other authors consider that this is all the same with a wide distribution. And also, as I mentioned, they are usually big plants, so we usually face incomplete herbarium specimens. This is even more concerning when you are facing a type, which is only a pinnate. Also, another problem is that it has this wide distribution among different continents, so there was no global study of it, just regional studies of the genus. And we didn't have a complete phylogeny. The most complete phylogeny was this one in the work of Gasper and collaborators in 2016, when they divided the family in so many genera, and with some additions of Barret, a French researcher. And as you can see, what these tree shows is a story of 2 delimited clades, one with the American species and another with the Austro-Pacific one, with the Africans mixed. But the posterior probability and bootstrap values were not that good. And it only covered 50% of the genus, with just 1 specimen per species. So what we wanted to do was to make a morphoanatomical study of all taxa, make a complete molecular phylogeny, and a taxonomical synopsis or a monograph. And I did it. I recently defended my PhD on September, 462 pages of pure fun. And how did we do it? Well, we basically used herbarium specimens because we were supposed to go to the field, but something about a pandemic happened. I don't know if you heard about it. But we got the chance to visit several herbaria and to ask for loans to several herbaria. And we also looked for some high-resolution images in all of these platforms. As a result, we could review 3,748 specimens, most of them in situ, and about 500 by images. And I have to add here the 394 specimens that I have just reviewed at field with my friend and colleague Sarah and Weston Testo, and the ones that I'm reviewing here, I can tell you there are a lot of them. So this is a summary of the characters we looked at in the specimens. We looked at the general morphology, but as you can see in this drawing, we also look at some more details, like the scales, the presence of aerophores, the anatomy of the fertile pinnae, or the palynology. The result was that we got the first global treatment for Parablechnum. We recognized 68 species, and we made a global identification key for them. We provide descriptions for all of the species we recognize with nomenclature notes, distributions, and other kind of notes like the etymology or how to distinguish them from close species. We also provide a plate for each species based on the herbarium material that we review. And we got to see some cool things, like, for example, we extended the distribution of some species, as you can see in the slide. For instance, Parablechnum marginatum was thought to be an endemism from Reunión, but we found specimens also from Mauricio. Parablechnum usterianum used to be a Brazil endemism, but we found also specimens from Paraguay, etc. We also found new species, but don't look at this slide really much because we still have to describe them. And as I said before, we made some more specific studies. For example, we studied the anatomy of the fertile pinnae because previous work showed that this is a character of high taxonomical importance in the Blacknaceae. So we made cross sections, which are the ones that you can see in the scheme in pink, and parasagittal sections in green. This is interesting because what our group has seen is that this cross-section of the pinnae is really distinctive in each genus, and you can easily distinguish the genera within the Blacknaceae by this. For example, you can see that the receptacle, the place where the sporangia are growing, are different. For example, it's really discrete in genera as Struthiopteris and Telmatoblechnum, or more elongated in other genera as Austroblechnum and Lomaridium. Also, the deepness and the presence of an abaxial growth is different. It can be more or less deep or not having one, like in Austroblechnum. And also, the indusium is really different among genera. We can have really simple indusia with just one layer of cells, as you can see, for example, in Telmatoblechnum, or really thick indusia with several layers of cells, as you can see, for example, in Struthiopteris. So we wanted to see if this variation can be seen also inside the genus Parablechnum. We saw that the general pattern of this anatomy is similar in Parablechnum. However, even though most of the species are dimorphic, there are some of them that are monomorphic. That means the sterile and the fertile fronts are all the same. So we made this division, and what we saw is that there are two kinds of monomorphism within Parablechnum. We have normal monomorphic plants with a flat leaf surface, but we have some other plants that the sterile and the fertile pinnae are all contracted. So we consider that this is monomorphic because they all look the same, but they are all contracted. I love these, they look like little aliens, by the way. Well, we also saw that in general the indusium is thick with several layers of cells, what we call a complex indusium. And we also saw that there is a variation in the number of vascular bundles in the costa, ranging from 2 to even 5 in Parablechnum umberti, for example. And we found another cool thing, that is this circunendodermal bundle around the vascular bundles of the costa in some species, as Parablechnum caliodontum, Parablechnum corvasoni, or Parablechnum nesophyllum. This structure is normally found in the petiole. It's made of sclerenchyma, and it gives like more thickness to the structure. But in some species of Parablechnum, they come all the way to the costa, and we don't really know why, but it hadn't been described before, as far as we know. We also made these parasagittal sections of the pinna, of the fertile pinna, to see how the veins looked. Well, it's not really intuitive, but in the upper side you would have the sporangia, and the addaxial side is in the back. Sorry, is in the lower part. So what we saw is that where the sporangia are, we have some veins that we call commissural veins that are small. They are wide, and they are all over the place, irrigating the sporangia. And in a perpendicular plane, we have what we call the lateral veins. Well, this is at the commissural, and these are the lateral veins in a perpendicular way. This is the ones we see here. Which are longer and less wide with another structure, and they all connect by an arc. This is what De La Sota Laborio drew in the 60s, And it has been discussed if the pattern within the Blachnesi was like this or not. And what we saw is that at least in Parablechnum, apparently it is. We also look at the spores at the optical microscopy, and we could measure them here with the equatorial diameter and the polar diameter. So we provide a measure for all of the spores. And we also made an electronic microscope study to see the ornamentation. We also looked at the sporangia. As you can see here, there are plenty of characters that we can see and measure and count within the sporangia. And before this, the most complete work of spores in the Blachnesi was made by Moran here in the audience and collaborators in 2018. And it was a work focused on the whole family. And regarding Parablechnum, they looked at 30 species, more or less 45% of the genus. And they described the spores of Parablechnum like reticulate cristate with filaments all over the place. We got to expand this sampling with 52 taxa, more or less the 80% of the genus. And we saw that the variation is higher. We could define 3 different groups. The group 1, which is really rare, is not reticulate. It looks like this. It only has 2 species. The group 2 with 4 taxa, that is cristate reticulate but don't form areolae. And group 3, which was the one that described Moran and collaborators in 2018. But within this group, which comprised most of the diversity of the genus, we also have variation. We have these cristate reticulate spores with a lot of filaments. But we also have species that don't have filaments, and a species which has, in addition, this rugulate perispore. And a lot of questions came to our minds when we saw this. For instance, is the ornamentation related to dispersion? We really don't know, but here in Parablechnum, we think we have a great model of study because we have a species with a similar distribution range with really different spore ornamentation, and right the opposite. A species with a really different distribution range, but really similar ornamentation in the perispore. Also, knowing the dimension of the spores is important because, as Barrington and collaborators saw, it's highly related to the genome size and the ploidy. And this is something that we still don't know a lot of in Parablechnum. These are the chromosome numbers known within the genus. We only know 8 of them, and there are 68 species. So there's still a lot of things to do here, and knowing the size of the spores can help. Another fun thing that we found is that there is a high percentage of broken and abortive spores in all of the specimens, not only in the hybrids. This is weird, but we also have seen it in another fern genera of the Blachnesi, like Struthopteris and Spicantopsis. And we don't really know why this happens, but it happens. This also has been useful taxonomically. As you remember, we've been talking before in the introduction about the complex Parablechnum cordatum, and that there was discussion to see if these were all the same species or different species. When we looked at the spores and the sporangia, we saw that they are really different, so we could assume that they were indeed different taxa. And among the sporangia, we found in some specimens, in some species, these paraphyses that are sterile structures that are mixed with the sporangia, and we don't really know what they are for, if they have a function. We even found a special type of paraphyses, what we call sporangiasters. These are sporangia that develop abnormally, forming this weird and funny sterile structure among the sporangia, which are more lower. And we don't know, as I said, we don't know what the function is for these structures. But what we've seen that is interesting is that the species that have them, usually they are all from the Austro-Pacific region, and they usually live on islands, and they don't have a great distribution range. So maybe they are benefiting the not the long-distance dispersal, but the opposite. And about the phylogeny, we also made a phylogeny. This is, again, the previous phylogeny that we had from Gasper and collaborators with these two clades, one from America and another one from the Austro-Pacific, with the Africans mixed. And as you can see, low support. So we used the same 3 chloroplast markers that they used, adding one more, the MAT-K. And we got 582 different sequences, most of them new, covering the 80% of the genus. We made a lot of different trees, both based on Bayesian inference and maximum likelihood, and we pruned our dataset, deleting those specimens with more missing data. And on summary, this is what we got. As you can see, it's not a really well-supported tree. Indeed, we have a backbone with a lot of polytomies, and there are species that appear in different clades within the tree. We only could see 3 clades that were repeated in all of our different phylogenetic trees. We were not really original. We call them clade A, clade B, and clade T. But what is interesting is that in clade A and clade B, we don't see a geographical structure. The dots are the region where the species grows, and as you can see, the Austro-Pacific, the American, and the African species are mixed in those clades. The only clade that did have a geographical structure was the clade C, with only species from the Austro-Pacific. So apparently, this previous scenario in which we had 2 well-differentiated clades geographically might not be right. And also, we made a divergence time estimation using secondary calibration points from another work that was recently published in 2022. And we saw that the origin of the genus is 38.3 millions of years ago, more or less. And this is interesting because if you see the distribution of Parablechnum, it is similar to a Gondwanic disjunction, but 38 millions of years ago, the continents were already separated. So what we think that happened is that Parablechnum got the current distribution by several events of long-distance dispersal. Indeed, in this work that I mentioned before, in 2022, we made a biogeographical history reconstruction of the whole family, and apparently the origin of Parablechnum would be in New Zealand and had reached the rest of the places from there. And we wonder why we had this low resolution in our tree. So we have several hypotheses. On the one hand, we believe that maybe we are facing a rapid radiation. We have to remember that the Blechnaceae is a really recent fern family which has a really high speciation rate. So this is what could be happening in Parablechnum. Indeed, in our day tree, we see that there is an American radiation with several cladogenesis events that coincide in time with the Andean uplift. This is a geological event that has led to a lot of different rapid radiation, both in plants and animals. So this could be an explanation. And also about the fact that we have species that appear in several clades all over the tree, we believe that maybe we are facing a case of reticulate evolution and cryptic speciation. And to explain this, I like this example of a work of the 90s that Chambers and Farran made in the Blechnum procerum complex, right now Parablechnum procerum, which is, it was a species that grows in New Zealand and had a really wide plastic diversity. So they studied this complex, and that way what they saw is that it was not just one species, but 5 different species in New Zealand. And also they look at the chromosome counts and the morphoanatomy, and they saw that all of these species hybridize. So if in a small group of plants in a relatively small area as New Zealand, we find this high percentage of cryptic diversity and reticulate evolution, what couldn't we find if we look at the whole genus? So what now? Now we would like to focus on the species complexes that we have detected in our phylogeny. We have to go to the field, and we would like to perform genomics analysis to see if we get a better resolution in our tree. But for this, we need funding. But fortunately, we got it. We recently got a grant from the Spanish government to work on this and to make this genomic analysis. and we are going to be able to go to the field. Indeed, we have already gone to the field. We went to New Zealand and New South Wales in Australia. We got a lot of fun, we made a lot of collections, but we were a little bit worried at the beginning, thinking, wow, we made all this way through the other part of the world. Will it be difficult to find Parablechnum? Well, in the first road slope, it was full of it, so it was pretty neat, indeed. We got to see all the species of Parablechnum recognized right now in New Zealand, the ones I have just told you about, which are wonderful, as you can see. And we also found all the species of Parablechnum that grows in New South Wales. So we are pretty happy. And what we are going to do now is we are going to use the Go Flag project. I'm not going to explain that because Sarah did it already. And when a new specimen enters in the project, we are going to perform high-seq workflow, and we are going to make a plastid phylogeny and a nuclear phylogeny. For the nuclear phylogeny, first of all, we are going to check if there is if we suspect that the specimen has an hybrid origin by paralogy warning. If we do find high paralogy and an allopolyploid origin is suspected, we are going to go to the other phase of the plants. As we are going to have the spores, we are going to try to culture gametophytes. And first of all, we want to describe the gametophytes because most of the gametophytes within Parablechnum are unknown. We only know the gametophytes of one species, so we want to see how they look like. And we also want to use the gametophytes to make the nuclear phylogeny in those cases in which we suspect that there is an hybrid origin. The reason why is that, as you may know, the gametophyte is the haploid phase of ferns. It has half of the genome. So when we do high-seq and high-piper, we have a lot of different loci, and they are from two different subgenomes because it's an hybrid, right? So we perform a workflow and we see that the different subgenomes, what we still don't know, which loci belongs to the subgenome A and which loci belongs to the subgenome B. And there is now a software called Homologizer that by Bayesian inference we can think which is the most probable scenario, which is the most likely combination. If we use the gametophyte with a low genomic charge, we believe that the computational workflow will be easier. We don't know yet because this has never been done. I will tell you about it in a couple of years. We are already cultivating this gametophyte. This is my student Guillermo, and we are culturing the New Zealand and Australian ones. They are so pretty. And coming back to the beginning, taxonomy in the 21st century. Why? Because taxonomy is important, because taxonomy is not an old-fashioned science. It's something that brings a lot of novelties, and it's not only made by all men in black and white pictures. It's even done by women sometimes. So I have a lot of people to say thank you to, but of course, thank you all for listening. And I will be happy to answer your questions if I know the answer.